Got it? So we use phenolphthalein in the experiment and not phenolphthalein. Phenolphthalein is formed after it gets oxidized. So phenolphthalein, on the other hand, can be made by oxidizing phenolphthalein. So what you see here is that when we put heme in the blood, it reacts with the Fe4 plus ions, reacts with phenolphthalein, which is colorless. In the presence of hydrogen peroxide, it will turn to phenolphthalein, which is pink, and the Fe4 plus ions will be converted to Fe3 plus ions. Okay, got it? Now what is the work of hydrogen peroxide here? तो अब इसमें क्या है, अगर आप देखोगे how is it done? So there is any kind of blood stain, you pick up that blood stain with a swab containing uh, uh, water, sterile water. Okay, you rub it, and then you add a few drops of phenolphthalein. Okay, at this stage it should not turn pink because you have not added H2O2. What is the work of H2O2? H2O2, the hydrogen peroxide, will be converted into water and oxygen by the peroxidase of blood. So without adding H2O2, there should be no color development. Okay, so that is what it says, that you take the sample, you add phenolphthalein, there should be no pink color at this stage. It is only after that you add hydrogen peroxide, the heme group will convert hydrogen peroxide to water and oxygen. The oxygen will go to your phenolphthalein, and it will, or you can say that because of the release of oxygen, phenolphthalein will be oxidized to phenolphthalein, and only then you will see a pink color. And this pink color should be generated within 10 to 15 seconds. Okay, so if the color generation is after 30 seconds, it is considered to be an invalid test. Okay, no, a pink color is a positive result, indicates the sample is presumptively positive. Acha, what is a presumptive test? What is a confirmatory test? Presumptive is that as soon as you reach a crime scene, you see something red which is splattered all over. You might say that okay, this is blood, but you are not confirmed, you are not sure. So first you do a preliminary test, only then you do a confirmatory test, and finally you do a individuality test. Okay, so no color development अगर pink नहीं हुआ after addition of H2O2 is a negative result, indicates that the sample is presumptively negative for the presence of blood. So how do you read the result? Presumptive positive, meaning it is not a conclusive test, but yes, it can be blood. Waiting for a period of time above 30 seconds will result in most swabs turning pink. So if you wait for more than 30 seconds and after that your swab is turning pink, that means there can be other things in the environment which is actually turning the phenolphthalein to phenolphthalein, and they are getting oxidized, it is getting oxidized on its own in the air. So that is why you do not rely on results which come after 30 seconds. Okay, so certain chemicals and biological fluids that do not contain blood can also cause the color change. So what can cause, so these are all viva questions now, what can cause a false positive result? Why is it a presumptive test? Why can't you confirm? Why is it not a confirmatory test? Because it can have a lot of false positives. So these substances have to be avoided since they produce a false positive result, and that is a positive result in the absence of blood. What is a false positive? That even when blood is not there, you get a pink color, that is a false positive. What can be the reason for false positive? First reason, maybe the result was noted after 30 seconds. Maybe there were other contaminants in the swab itself which could oxidize phenolphthalein to phenolphthalein. So see, anything which oxidizes phenolphthalein to phenolphthalein is going to give you this test. Okay, so there can be other chemicals, other biological fluids also which give this test. So what are the limitations? So like I said, it is presumptive, it depends on the peroxidase activity. So this test cannot differentiate between animals and humans. So if you see a blood splatter, right, if you see a blood splatter and I ask you to determine the species of the person, whether it's a human's blood or a cat blood or a dog's blood, you will not be able to distinguish, because the main chemistry behind is that hemoglobin will break down hydrogen peroxide, and it will the oxygen will then oxidize your phenolphthalein to phenolphthalein. So the blood is the same, the hemoglobin is same in animals as well as humans, so both animal and human blood will give this test as positive, so it cannot differentiate between animal and human blood. Then there can be insufficient sample quantity, development of positive reaction, कम है sample तो positive कई बार नहीं आएगा. Color change must be observed between within 15 seconds, and many a time there can be color development before addition of hydrogen peroxide also, and certain plant peroxidases also react similar to blood in catalyzing this reaction. So typically these stains are associated with plant tissue, can be visually distinguished from blood, but plant peroxidases, they tend to be unstable, losing their ability to oxidize phenolphthalein reagent over time. So some plant peroxidases can also give you a cross reaction with this test, but it is seen that if you keep these for a longer period of time, their color also differs and they also lose their peroxidase activity. Okay, next test is the TMB test. किसी को phenolphthalein test में कुछ पूछना है? Phenolphthalein test is also known as the Kastle-Meyer test, ना कंफ्यूज नहीं होना है. प्रिजम्पटिव टेस्ट ब्लड का Kastle-Meyer टेस्ट है, phenolphthalein टेस्ट सेम है. Then TMB test. TMB test is basically an improvement over the benzidine test or the Adler test. Adler test पहले यूज होता था, उसमें जो रिएजेंट यूज होता था बेंजिडीन, वो कार्सिनोजेनिक था, इसीलिए उसका एक डेरिवेटिव देखा गया, टेट्रा 3355 33 प्राइम 55 प्राइम टेट्रामेथाइल बेंजिडीन, which is non carcinogenic, and then that is used. Okay, so similar to that of the previous test, here also we are utilizing the peroxidase like activity of hemoglobin. When hemoglobin, TMB, and an oxidizing agent like hydrogen peroxide are combined, the TMB is oxidized, causing a visible change from pale blue to deep blue color. Okay, so this is what it shows, that this is TMB, which is colorless. So you have take a swab, you add TMB, and then you add H2O2. H2O2 will be acted upon by the heme, which has a peroxidase like activity, and this will lead to reduction of your, oxidation of your TMB to tetramethyl benzidine diamine. So when it forms this diamine here, this is blue green in color, this is pale blue, colorless to pale blue in color. So earlier it was colorless to pale blue, turns to dark blue or bluish green in color. Okay, so this is, I am not explaining this. What are the limitations? So many of the fruits and vegetables can give you a positive test for TMB. Most of the fruits, vegetables produce false positive test for TMB. Some of the vegetables produce false positive color, like asparagus, avocado, green beans, broccoli, cabbage, capsicum. All these can give you a false positive result with TMB, and they are all coming in within that 20-second range. So the test is again studied within 20 seconds. So they are all coming in between 20 seconds. And again, a number of other fruits can also give you positive result. And if you wait for a minute, even red grapes can give you a positive result with TMB. What is the advantage? That it is highly sensitive. All the reagents are non carcinogenic, easy to perform. So, no need for a confirmatory test if the test result is negative. So, it is usually used as a good indicator of a negative test, क्योंकि false positive तो बहुत सारे आ जाते हैं, पर false negative बहुत कम आते हैं. So, it can be used for, if it comes as negative, then you are sure कि हाँ blood नहीं है. ठीक है, and it can be made and used for up to 1 week. देन confirmatory टेस्ट. अब confirmatory टेस्ट कौन से थे ब्लड के? टाकायामा एंड टाइकमैन टेस्ट. टाकायामा टेस्ट we have not done, we have just studied, and Teichmann we have done in the class. So what are these tests? Both of these tests are relying on the capability of blood to form specific crystals. टाकायामा में अलग तरीके के क्रिस्टल्स बनते हैं, टाइकमैन में अलग तरीके के क्रिस्टल्स बनते हैं. So टाकायामा Microcrystal confirmatory test for blood. It is frequently used in forensic labs, also called as the hemochromogen test. And a positive result is indicated by pink feathery crystals of pyridine hemochromogen. So, what exactly is hemochromogen? So, this test is performed in the presence of alkali, first of all. So, hemochromogen, also spelled as hemochromogen, is a compound of heme with globin modified by the action of alkali. The crystals of the crystals of hemochromogen are prepared by heating the blood with Takayama's reagent. So आपको reagent डालना है, आपको heat करना है. तो जब आप heat करोगे, red blood cells rupture होंगी, hemoglobin निकाले, उसमें से hemoglobin निकलेगा, and they will form ferrohemochrome. Now, during the process, the ferrous atom, the ferrous form of the iron is converted to the ferric form due to the presence of the base in Takayama reagent, which is NaOH, and so it forms hemetin. Okay, so when the ferrous form converts to the ferric form, it forms hemetin, alkaline hemetin. Now, this alkaline hemetin will combine with the second thing which is present in the Takayama reagent after NaOH, it is pyridine. So hemetin will combine with pyridine. What is pyridine? It's a nitrogenous compound, and when it combines with pyridine, it forms insoluble colored crystals. And these colored crystals are of pyridine hemochromogen. Okay, so hemochromogen pehle kya tha? It was that when it is heated in the presence of alkali, the RBCs will rupture, the hemoglobin will come out, but it will still remain attached. So it forms ferrohemochrome. And then when it converts from ferrous to ferric state in the presence of NaOH, it forms hemetin. This hemetin, when reacts with pyridine, will form pyridine hemochromogen, which appears as pink colored crystals. Now, in this reagent, saturated glucose is also added as a reducing agent in the reaction, which reduces the solubility of hemochromogen and forms numerous crystals. So, this is what you see here. This is hemoglobin. When you add these three things, all these three form the Takayama reagent, they will form pyridine hemochromogen. So, now you see these are bound to the nitrogenous pyridine, which is here. So, these are forming the pyridine hemochromogen. How do you prepare it? You add these things, not necessary for you to. So, how do you do the test? You add 1 to 2 drops of blood, spread it evenly, add 2 to 3 drops of Takayama reagent. What does it contain? Glucose, NaOH, pyridine, distilled water. Okay, then you heat it for 10 to 12 seconds and you see. So, these are needle shaped crystals, and these are pyridine hemochromogen known as pyridine ferroprotoporphyrin. Okay, so they can be termed as Takayama crystals, they can be termed as pyridine hemochromogen crystals, they can be termed as pyridine ferroprotoporphyrin, because this structure here is of pyridine ferroprotoporphyrin. Okay, this is how it looks under the microscope. All these are different advantages of the Takayama test. Now, it is best for old stains. It is more reliable than the other test we'll study. Very small amount of sample is required. The reagents are non-corrosive. Many a times heating is not required. Even in the absence of heating, it will form these crystals, and it can develop hemochromogen crystals even if blood is exposed to fresh water decomposition media for 30 days. So even if blood is present in freshwater areas or somewhere there are some stains are present, still it can form these. What are the disadvantages? It takes a little longer to perform. For full surety, examiner will have to wait for 30 minutes in order for the crystals to form. Okay, and therefore sometimes it is not prescribed in the uh, current protocols. The one that we have studied is the one we have done in lab is the Teichmann test. So in Teichmann test, it is based on the reaction of the heme part of blood with NaCl and glacial acetic acid to form brown colored rhombic crystals. Now these rhombic crystals are brown in color and they are of hemetin chloride. So they are ferriprotoporphyrin chloride crystals. वहां क्या था? These crystals were pyridine ferroprotoporphyrin, whereas here the crystals are of ferriprotoporphyrin chloride. Okay, so generally, if you look at the structure of hemoglobin, it has six binding non-proteinous sites. Four have nitrogen coordination bonding to tetrapyrrole ring with Fe2 plus, while the fifth bonding is between iron atom and the nitrogen of the deprotonated histidine. Now, when NaCl with glacial acetic acid reacts with heme, a heme derivative known as hemetin is formed, with the central iron being in the Fe3 plus state. Okay, the following is the chemical reaction, which is the chemical reaction here. So, this state will be from Fe2 plus to a Fe3 plus state, and the chloride will attach here. Okay, so in the presence of glacial acetic acid will help to rupture the cell membrane, allow the protein to move out, the hemoglobin to move out. The NaCl will help in formation of these crystals. Okay, what was the procedure? The procedure, so it's a confirmatory test. Okay, the procedure is that you take the blood, you add a few drops of the reagent which is containing your NaCl and glacial acetic acid. You cover the slide, heat it for a few seconds, observe under the microscope. Now, Teichmann's reagent can have KCl, KBr, Ki, and acetic acid. Many times there are certain, you can say there are certain changes or modifications which use only one of these. Okay, so Nips reagent is one of the modification of the Teichmann's reagent. So usually Teichmann's reagent is glacial acetic acid in the presence of halides will give you these kind of crystals. So 1-2 drops of blood, 2 drops of Teichmann reagent, you heat it for 10 to 12 seconds, and then what do you see? You see brown rhombus crystal shaped of crystals of hemin, also known as ferroprotoporphyrin chloride. Okay, so that was, these are the crystals. So these are brown in color, they are not pink, they are brown in color, they will be rhombus shaped, and these are of ferroprotoporphyrin chloride. Now, false negative results. Why do you get false negative result? If the stain is old, that is why Takayama is preferred when the stain is old. Similarly, if the stain is washed or has been present in water, again it will not give you a good result. It will give you a false negative. If the stain was previously treated by some chemicals, again this Teichmann test will not give you good results. Okay. Now, if you're using concentrated, if you're not using proper glacial acetic acid, you will not get it. And if too much of sodium chloride crystals is added, you are not following the proper procedure of making the Teichmann reagent, it will be negative, false negative. And again, overheating will give you negative results. So it is only 25 to 62.5 degrees पे अगर आप करते हो तो ठीक बनते हैं crystals, otherwise charring हो जाती है and you will not be able to see anything. Modified versions of the Teichmann test, there are certain, the NIPS modified Teichmann test. So in this modified version, instead of using a single salt, NIP uses a combination of chloride, bromide, iodide. आप लोगों ने क्या यूज किया था? Can somebody tell me? फाइल में क्या लिखा है?
Ma'am, NIPS reagent.
हाँ, उसका क्या लिखा है composition?
Potassium iodide, bromide, chloride, glacial acid. Right?
So it is this one, right? So यहाँ पे I was a little confused. The earlier Teichmann's reagent only had chloride. Now the NIPS reagent, which is the modified one which we also used, it consists of 1% potassium chloride, potassium bromide, potassium iodide, and dissolved in glacial acetic acid. Okay, so that was about it. And अगर आपसे सलाइवा का पूछते हैं तो सलाइवा का बहुत ही सिंपल सा एक आपका इंटरप्रिटेशन है, एंड वो ठीक से लिखा भी हुआ है आपकी फाइल्स में. So it is why, what is the result? Will you get blue coloration if the saliva would be present? Can somebody answer?
No,
We-
ma'am.
No, because what will happen? the starch reacts with iodine to give you a blue black color. And in the presence of saliva has salivary amylase which will break down the starch, and amylose and amylopectin do not give a blue color in the presence of iodine, and therefore wherever saliva would be present, instead of a blue color you will see no color or discoloration. If earlier there was color, there will be removal, or the it will turn colorless. Okay. So next is hair analysis. Now hair is a trace evidence. So there are different types of evidence. Some are trace evidence, some are class evidence. So what are trace evidence? Definition: tiny amount of material that can be exchanged when objects or people come into contact. So there is a principle called the Locard's exchange principle, and it says that every contact leaves a trace behind. Okay, so those tiny things which are left from one object onto a person, or when a person meets a person, all these are trace evidence. For example, hair, fiber, glass, paint, soil, gunshot residues, all these are trace evidences, and they can help you to provide a link between the suspect and the victim. Then there are class evidence. So class evidence can be linked to a group of or a class of objects, but will not be you will not be able to pinpoint on a single source or a unique source. For example, if there is a certain paint mark found, so you can say, okay, this is the color of the paint, so it will help you to narrow down the circumstances where the crime would have occurred. So for example, automotive paint from a specific manufacturer, a brand of a new tire, or certain types of fibers. So अगर certain type of fiber मिले हैं, तो आप ये ढूंढ सकते हो कि अच्छा ये वाले fiber कौन सा टाइप का है, आप उस मिल में जा सकते हो, या वो वाले सर्टेन मिल्स कहाँ है उन जगह पे जा सकते हो, बट यू विल नॉट बी एबल टू फाइंड किस पर्सन के पास ये था. ओके. So why do we do hair analysis? Because hair can help you to link individuals and locations, because hair can transfer very easily from one person to other. So at a crime scene, hair can be present on a victim's jacket. The hair of a suspect can be present. So small, it can't be seen, but there are very fine hair which can be present. So hair can be easily physically transferred. Then again, why do we study hair, or why hair analysis is done? You can do a DNA analysis from that. So here I'll just show you. Hair can have roots as well as shaft. So the root can give you the nuclear DNA, the shaft can give you the mitochondrial DNA. If you have the root, then you can identify the individual. If you have the shaft, you can exclude a large number of people. So you know about maternal inheritance, uska pata chal sakta hai, who is the mother, who is the, so you get to know the lineage. You might not be able to find the exact person, but it will help you to exclude out a large number of people. Then microscopic comparison. So microscopic comparison can help you to know about the color of the hair, the length, the root appearance, determine the age, the gender, the racial origin. Okay. So this is the diagram of a hair. This is the hair shaft. Okay. So what you see outside mostly is the hair shaft, and inside is the root of the hair. Okay. So this is the hair bulb. So what does the shaft consist of? Medulla, cortex, and cortical. Then there's the sebaceous gland, or the oil gland which produces the oil. This is the inner root, the outer root. This is the hair matrix and the papilla. So this is the hair bulb. Okay, or you can say the follicle region here. This is the follicle region. So if the hair uh, is is there, so it can actually also tell you how the crime has been committed. If you are seeing hair with the root, that means it has been forcefully pulled, and there is some kind of a uh, uh, you know some kind of a fight which has taken place, or some kind of resistance which was there, and then the hair was pulled out. So it helps you to determine the what might have happened in the crime scene. So phases of hair growth. At which phase, so like I told you, that hair can help you to find out the age of a person, or not just the age of the person, many other things. So there are different phases of the So these are phases of hair growth. Do not confuse it with phases of our growth. Our growth and the hair growth is different, है ना? So anagen is the active growth phase, 2 to 6 years of the hair, not our 2 to 6 years. Okay, so anagen stage, active growth phase. Permanent hair removal can only occur during this active growth stage. This phase may last up to 2 to 6 years. So here is a actively growing phase of the hair, where the hair is healthy and it is actively growing. Then there comes a catagen phase or a transition phase, which lasts up to 1 to 2 weeks. So the club hair transition upwards, so it starts to move upwards, this portion towards the skin pore, and the dermal papilla begins to separate. So this is the dermal papilla here. The dermal papilla begins to separate and this starts to move upward. Usually lasts 1 to 2 weeks. And then there is the telogen phase. So telogen phase is the resting phase, about 5 to 6 weeks. So resting phase, the dermal papilla fully separates from the follicle, and it can, so this is fully separated from the follicle. That means that this is not an actively growing hair. It can shed. So this hair will be shed, but it can remain attached to the scalp for 5 to 6 weeks. So it is not that when somebody says that I've been experiencing hair fall for the heavy hair fall for the past 2 weeks, that it is in those last 2 weeks something has happened. It has happened much before that, during this stage. During the anagen phase only something has happened, that it has transitioned to the catagen and then to the telogen. So the telogen phase remains for about 6 weeks before the hair is shed. So when the hair is shedding, it is the telogen phase, and then there is a return to the anagen phase when the new hair starts to form. So the hair matrix will develop. So the dermal papilla moves upwards to meet the hair follicle. Hair matrix begin to form. So when there is a hair which is formed at a crime, it is found at the crime scene, what all has to be done? And please, I hope everybody is listening, because tomorrow you will get spot questions. You will get spot questions which are related to this. And if you hear properly, only then you will be able to answer. We have not practiced spots as such, but I am telling you the spots as while we were discussing in class also and right now also. Okay, so when hair is found at a crime scene, the first thing that has to be seen is whether you have got the hair root as well, or whether you have only got the shaft. Okay, so when you get hair, you can find the morphology. You can take mitochondrial DNA if it is the shaft. You can see the nuclear DNA if it is the hair root. Now, from the mitochondrial DNA, you can get the maternal analysis can be done. From the nuclear DNA, you can identify an individual. Now, there are other things like you can find the methylation, you can find the proteins attached, but we'll not go into much detail. Depends upon डीएनए मिथिलेशन से क्या होता है, मिथाइलेशन से आपका एक्सप्रेशन ऑफ जीन्स पे फोकस होता है, मतलब एक्सप्रेशन ऑफ जीन्स वेरी करता है. सो हर एज के साथ ये एक्सप्रेशन ऑफ जीन्स will vary. How much of methylation is there will help you to infer the age. Okay, so this is a hair which is with the follicle. Okay, so whenever you are seeing hair which has these dark colored tips over here, that means these are hair which have been pulled out and these have roots with them. Okay, so this is microscopic view of it. This is how it will appear otherwise, if you see it otherwise under a magnifying glass. So this is pulled hair. So you see the dark color is there, but because it was pulled forcefully, or it was just pulled by a person just sitting like this, so the tip is, or the root is a little distorted here, then forcibly removed. So you see the bulb which is otherwise shed. जब shed होता है तो ये bulb पूरा ऐसे proper form है. जब forcefully है तो ये irregular हो गया है थोड़ा. So this is a forcibly removed. So the point that I'm trying to make here is that wherever you will see these kind of, if you see these kind of dark dots at the end, that means hair has been pulled out. Okay, now if there is only shaft available, there is no root, then it can tell you how the crime was committed. For example, if the hair was burnt or there was charring, you will see this kind of a structure. If it was cut, then you see a cut in the shaft. If there was a razor which was used, then you will see a slant cut here. And if the hair is split, when the hair split or there are split ends, so you see this kind of a structure. Okay, so this is what a split end looks like. Then the race of a person can be determined. If there is a hair which is found at a crime scene, one can determine whether it is from an African person, an Asian person, or a European person if you look closely at the hair. Acha, how will you find out if at a crime scene some hair is found? How will you find out if it is human hair or animal hair? One of the first thing that has to be seen is the pigment. Pigment in human hair is the same throughout. Okay, mostly it remains constant in color, and the pigment will be present. Pigment कैसे पता चलेगा? अब ये जो आपको ये जो दिख रहा है, तीन चीजें, सबसे बाहर आपका क्यूटिकल होता है, उसके बाद कॉर्टेक्स, और ये जो आपको ब्लैक कलर का दिख रहा है, दिस इज मेड्यूला. So in the cortex you can see small dots of melanin. So that will be the pigment. So अगर किसी का white hair है, उसमें आपको pigmentation बहुत कम दिखेगा, किसी का black hair है तो ज्यादा दिखेगा. And humans के pigmentation uniform होता है. Animals के hair में अचानक से आपको कोई pigment दिखेगा, कभी नहीं दिखेगा, फिर दिखेगा, फिर नहीं दिखेगा. So shows rapid and radical color changes in case of animal hair. In case of human hair, there is a constant coloration. Then the most important part, in case of humans, medulla is very thin. So the black line here which you see is the medulla, is very thin, that, and many a times it is absent and it is fragmented. ये जो ह्यूमन हेयर है इसका बहुत अच्छा मेडुला है, किसी के बहुत हेल्दी हेयर है. सो otherwise the human hair has fragmented medulla, it is a very thin medulla, and it is many a times it can also be absent. Okay, so what it shows here is the medullary index. Medullary index is your medulla width upon the hair width. What is the diameter or width of the medulla upon what is the width of the hair? Okay, so in case of animals this medullary index is high, it is 0.5 or more. Why? Because the thickness of the medulla is high. Okay, so the medulla is very wide, it is regular and well defined, so they will have a proper medulla. And the medulla also has patterns. These medulla, for example in cat, it is a stacked medulla. अगर आप ध्यान से देखोगे cat का, तो आपको छोटे छोटे डिस्क दिखेंगे जो साथ में stacked हैं, right? So that is how you can differentiate between the animal and human hair with respect to medulla. Then comes scale. The scale has a overlapping imbricate flattened scales हमारे केस में है, और coronal scales या crown shaped spinous shapes उसमें है. So you see here, अच्छा, this is showing the medulla. अलग अलग मेडुला दिख रहे हैं आपको, है ना. ह्यूमन के केस में इसमें है ही नहीं वो मेडुला, एंड माउस के केस में आपको स्टैक्ड दिख रहा है, स्क्विरल का अलग है ना, सब में अलग अलग मेडुला है. अच्छा, देयर इज लेट मी शो यू. I think I wanted to show some pictures, so I have put this slide here. So cuticle is on the outside, cortex is the inner layer, medulla is the innermost layer. The cuticle, which I wanted to show you. So in case of humans, the cuticle is an imbricate cuticle. Whereas in case of animals, it can be coronal or it can be spinous, flower-like. Okay, so the scales can be different in case of humans and animals, and that is how you can differentiate between them. So human scales, this is what human scales look like. So they are imbricate. Okay, so cuticle scales of animals, cuticle scales of humans. So cortex has the melanin pigment. I've told you, in case of humans the pigment is more or less same. In animals there will be radical changes in that. Medulla, again this is showing you a continuous medulla, fragmented, interrupted medulla, solid medulla, no medulla. ना. Human medulla can be continuous, fragmented, or absent. Most of the times it is either fragmented or absent. In animals, there can be different types of medulla. Okay, this I've told you, this I've told you. Yeah. So this shows a hair which has fallen out, and this shows a hair which has been forcibly removed. You can get DNA from the hair, that also I've told you. And hair is also a good trace evidence in order to know about toxicology. You know, many a times chemicals that are ingested or absorbed by the skin can be detected in the hair shaft. Hair में आपको आप क्या खा रहे हो या क्या chemicals आप ले रहे हो, उसका effect आपको hair में मिल जाता है. And forensic scientists can actually they have studied the effect of different chemicals on the hair, and they will give you different results. Okay, now why hair is a good sample for studying? Because it is easy to collect and store, है ना. It is externally available. आपको कोई extra ऐसा नहीं है कि कुछ विसरा या कुछ बॉडी को आप पहले ऑटोप्सी करोगे फिर निकलेगा, है ना. And it can give you a history of the use of drugs or poisoning, and it remains as it is. The level of degradation, it is very resistant to effect of chemicals, it is very resistant to degradation by environmental factors, so the hair can remain like that for a long period of time, and that is why it is a good evidence. Getting it, everybody?
Yes, ma'am.
आगे पढ़ाऊं या बस करूं,
मैम पढ़ा दो.
या टाइम वेस्ट हो रहा है आप लोग का? अच्छा, ज्यादा मुझे नहीं पढ़ाना है आगे, बाकी तो आप खुद भी पढ़ेंगे ना कुछ. अच्छा, ब्लड स्प्लैटर एनालिसिस. ब्लड स्प्लैटर एनालिसिस विल टेल यू हाउ द क्राइम माइट हैव अकर्ड. ठीक है, क्या वेपन यूज हुआ, कि गन यूज हुई, कि नाइफ यूज हुआ, है ना. इट कैन टेल यू कि सस्पेक्ट कहाँ खड़ा हो सकता था, विक्टिम कहाँ था, कहाँ से एंट्री हुई सस्पेक्ट की. सो ऑल दीज थिंग्स ब्लड स्प्लैटर एनालिसिस कैन टेल यू. अब ब्लड स्टेन्स कितने टाइप के होते हैं? पैसिव, ट्रांसफर एंड प्रोजेक्टेड और इम्पैक्ट स्टेन्स. पैसिव स्टेन्स मीन्स जब ब्लड जो है अंडर द इफेक्ट ऑफ ग्रेविटी वो अपने आप गिर रहा है. लाइक इफ देर इज अ कट एंड यू पुट योर फिंगर, नीचे को अगर कर देते हैं तो ड्रॉप 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 जो फ्लो होता है, दैट इज अ पैसिव स्टेन. सो ड्रॉप्स, फ्लोज, pools of blood are all passively formed under the effect of gravity. Then there are transfer stains. अगर किसी ने कोई मर्डर किया और वो वहां से चल के गया तो उसके जो फुटप्रिंट्स बनेंगे, those would be transfer stains. Okay, so whenever there are, so transfer stains will leave wipes, swipes, pattern transfers behind. And then there are projected or impact stains. जब किसी चीज से मारते हैं तो there will be a splatter. For example, there can be a gush of blood, there can be splashes, there can be arterial spurts. All these are splatters. Now, how does that occur? अच्छा, सबसे पहले blood का हमने एक स्पेसिफिक डेंसिटी, जब हमने आर्टिफिशियल ब्लड बनाया था, एक स्पेसिफिक डेंसिटी को अचीव किया था, उसके बाद ही एक्सपेरिमेंट किया था. व्हाई इट इज नेसेसरी? बिकॉज अदरवाइज you will not get those specific angles, you will not get the specific pattern. It is only because when blood has a certain surface tension, it has a certain viscosity, that it is able to form those patterns, and those patterns can be studied. One of the experiments that we did was to see the effect of angle, है ना, the angle of impact. So angle of impact, or theta, so it is the formula is sin theta is equal to width versus length, है ना. सो एंगल ऑफ इम्पैक्ट, ये है एंगल ऑफ इम्पैक्ट. अब एंगल ऑफ इम्पैक्ट कैसे देखते हैं? साइन थीटा इज इक्वल टू width upon the length, width upon the length. So theta will be sine inverse of width upon the length, and that will help you to find the theta or the angle. Okay, so as the angle 90 degrees पे अगर कोई ड्रॉप गिरी है, मतलब यहां से अगर कोई ड्रॉप गिरी है, इट इज ऑलमोस्ट राउंड. अगर वो ग्लास सरफेस पे गिरेगी, ग्लास सरफेस, स्मूथ सरफेस पे गिरेगी, तो उसमें ये जो सैटेलाइट प्रोजेक्शंस हो रहे हैं छोटे छोटे, वो नहीं होंगे. लेकिन अगर वो किसी रफ सरफेस पे गिरेगी, जैसे कि पेपर, तो उसमें जितना ज्यादा रफ सरफेस पे गिरेगा ड्रॉप, उतने ज्यादा उसमें वो सैटेलाइट प्रोजेक्शंस होंगे. सो जब आप एक कोई ब्लड स्प्लैटर देखते हो, तो आप उसमें एनालाइज ये भी कर सकते हो, अगर वो एकदम राउंड है तो वो वर्टिकली 90 डिग्रीज पे गिरा है, अगर किसी एंगल, अगर ये देखो जैसे एंगुलर है, so if it is angular that means that is at a certain angle, it is at a certain angle, ये 10 डिग्रीज पे है, ये इलोंगेटेड है ना, तो अब ये जब इलोंगेटेड है तो आप इसमें वही फॉर्मूला लगाओ, sin theta is equal to width upon height and you will find out the theta by using the table. देंगे नहीं आपको ऐसा करने को, बट अगर करा जाए तो then what is the height? So with the height the डायामीटर विद इंक्रीज, जितना छोटी जगह से ब्लड ड्रॉप होगा उतना छोटा ये बनेगा, जितना ऊपर लेते जाओगे उंगली उतना ज्यादा बड़ी ड्रॉप बनेगी, ठीक है। देन व्हाट आर द डिफरेंट टाइप्स ऑफ ब्लड स्प्लैटर? अब अगर इस तरीके का ब्लड स्प्लैटर है दैट मीन्स कि कोई आर्टरी कुछ तो स्लिट करी गई है किसी पॉइंटेड ऑब्जेक्ट से या किसी नाइफ से, द आर्टरी हैज बीन cut because of which suddenly there is a gush of blood. This is also a gush of blood, arterial है। then there are transfer patterns, ये आपको एक हाथ दिख रहा है जो इसमें ब्लड लगा था वो कहीं पे रखा गया, so this is a transfer pattern. then these are cast off patterns, कि जिस चीज से मारा है ना, जिस चीज से मारा जब वो चीज वापस उसका उसके इम्पैक्ट से उस चीज से वो पीछे वाली चीज पे बना है पैटर्न, ओके। जस्ट अ मिनट। okay, so I've told you angle, इम्पैक्ट, हाँ इम्पैक्ट कैन बी फॉरवर्ड स्पैटर हो सकता है, बैकवर्ड भी हो सकता है। बैकवर्ड बहुत बार कास्ट ऑफ पैटर्न में बनता है। इम्पैक्ट स्पैटर लो वेलोसिटी, मीडियम वेलोसिटी, हाई वेलोसिटी का अलग अलग बनता है। अगर किसी का गन शॉट वाला है तो वो हाई वेलोसिटी है, उससे एक मिस्ट बनेगा। सो फॉर एग्जाम्पल हियर यू कैन ओनली स्विंग द हैमर सो फास्ट बाय हैंड। एक हैमर जो है उसको आप, आपका हाथ कोई मैकेनिकल वो तो नहीं है ना, कि मशीन तो नहीं है कि एक बहुत ज्यादा फोर्स है, एक सर्टेन अमाउंट ऑफ फोर्स से ही हैंड से लग सकता है। सो इट इज अ लो वेलोसिटी। Similarly, if somebody is hitting someone with a pipe, again it is a low velocity. What would be high velocity? A gunshot wound. Okay, so there will be a fine mist particles all around. So agar misting hai, so this kind of a blood splatter is mist. So this is a fine mist of smaller particles occurring because of high velocity, mostly seen during gunshot. ये किस वजह से बना है? This is because of the low velocity. This is because of the low velocity. And high misting around, this is because of the gunshot. Now if there is another high velocity splatter, this shows a splatter of a suicide bomber who has detonated. So again this is a mist all around, ये पूरे में mist है, okay. So this is also high impact splatter. Impact high है but क्योंकि इतना blood है हम शायद it can be arterial spurt, पर arterial spurt ऐसे नहीं दिखता है, जो आपको पहले पिक्चर्स दिखाई थी वो है, ठीक है। We will not go into this कि कैसे निकालते हैं। There is a certain method by which you actually find out where a person might be standing and what was the height and कहाँ से गोली चलाई, वो सब चीज पे नहीं जाएंगे। Then whenever there's a gunshot splatter, there can be a fine forward spatter exit, जहाँ से exit किया, and एक back spatter भी आता है, only if the bullet does not exit the body. So there is a back spatter also. So many a times when there's a drawback effect, क्या होता है कि जब आप किसी को पास से शूट करो तो जो पर्सन है उस पे भी ब्लड के छींटे आए, या जो गन है उस पे भी ब्लड के छींटे आए, ठीक है। देन देयर इज कास्ट ऑफ, आई टोल्ड यू ब्लड कवर्ड ऑब्जेक्ट फ्लिंग्स इनटू एन आर्क, एंड जो पीछे चीज है उस पे पैटर्न आएगा, that would be a cast off pattern. Arterial spray, pressure of arterial pumping blood out of the injured area. Injury site begins the biggest spurt. Trail shows the victim movement. So arterial spurt होने के बाद जैसे जैसे कोई move करेगा उसका movement दिखेगा कि ये move किया है, इतना blood gushing out. So you can determine the victim's movement after the injury, especially in the case of a arterial spatter. Then expirated blood, अगर ब्लड नोज से कहीं से निकला है, सो दैट कैन ऑल्सो बी सीन, एंड इट बेसिकली शोज, अगर बाथरूम में आपको या वैसे एक्सपिरेटेड ब्लड दिख रहा है तो मीन्स कि उसको किसी और तरीके से मारा गया है जिससे कि इंटरनल इंजरीज ज्यादा हुई हैं। Then there are void patterns. So like I think you've pasted the picture. अगर scissor बीच में रखी और so there is a void या खाली हो जाता है उस जगह से, so it takes the shape of that object. So उतनी जगह पे कोई blood नहीं होगा, बाकी उसके आसपास होगा. Then contact transfer patterns I've discussed already. So these are transfer patterns. Shoe का हो सकता है, hand का हो सकता है. Now there can be flow, passive movement of the blood. So large amount of drops can be there. There can be pools of blood beneath the body where the body is lying. This is telling you the angles. Skeletonized pattern kya hai, which is telling you the shape of the object. So edges of the stain dry to the surface. Okay. So this was about blood splatter. एंड फिंगरप्रिंट एनालिसिस. अच्छा, एक तरीका क्या है, ब्लड स्प्लैटर जो वैसे नहीं आपको दिख रहे हैं, वाइप कर दिए गए हैं क्राइम सीन से, उनका क्या तरीका है देखने का, ल्यूमिनॉल स्प्रे करके. सो ल्यूमिनॉल केमिल्यूमिनेसेंट मेथड है. ल्यूमिनॉल विल रिएक्ट विद द ब्लड्स हेमोग्लोबिन and it will give a glowing effect, but it can only be seen in the dark and one has to quickly take the photograph in order to see the blood. So this is showing you that the blood stains were wiped off. Until unless अच्छे से साबुन से लगा के या कोई केमिकल स्पेसिफिक केमिकल्स डाल के ना क्लीन किया जाए, जो फिंगरप्रिंट्स हैं, जो ब्लड स्टेन्स हैं, वो नहीं जाते हैं. देयर विल बी ट्रेसेस ऑफ द ब्लड स्टेन्स व्हिच कैन बी सीन बाय दिस ल्यूमिनॉल स्प्रेइंग. देन फिंगरप्रिंट्स कौन कौन से टाइप के होते हैं, लेटेंट, पेटेंट एंड प्लास्टिक. लेटेंट आर दोज व्हिच आर इनविजिबल, पेटेंट आर दोज व्हिच आर विजिबल. फॉर एग्जांपल, आप लोग कोई स्टेनिंग करी और उसके बाद अपनी कॉपी पे हाथ लगाया तो पेटेंट फिंगरप्रिंट बन गया. प्लास्टिक फिंगरप्रिंट साबुन पे, क्ले पे, डो पे, वो जो फिंगरप्रिंट्स बनते हैं वो सब प्लास्टिक फिंगरप्रिंट्स हो गए. ठीक है. फिंगरप्रिंट डेवलप कैसे करोगे, पाउडर से. अब हमने सिर्फ ब्लैक पाउडर किया जो कि कार्बन पाउडर था, पर बहुत सारे तरीके के पाउडर्स आते हैं. देयर आर व्हाइट पाउडर्स, मैग्नेटिक पाउडर्स, ऑल ऑफ देम डिपेंड अपॉन वेयर यू वांट टू पिक अप द सैंपल फ्रॉम. Then there are chemical methods. Ninhydrin is there, silver nitrate method is there. There is also cyanoacrylate fuming. You can read these methods, all of them. The ninhydrin method is relying on which method? It is relying on the principle that there will be amino acids which will be present in the, when the sweat glands produce sweat, there will be certain cells or certain proteins also which will be released from them, and these proteins can react with the ninhydrin to give a blue color, because all amino groups, they react with ninhydrin to give a bluish color, humans purple, that is why you see them. So this is the ninhydrin test. Then non-invasive method of fingerprint development. So अगर आपको उसमें कोई powder या कुछ नहीं डालना है, so then there are alternative light sources. So there are alternative light sources or UV light. Under UV light also you can see the fingerprints, because again the tryptophan and the tyrosine, they will absorb light, the UV light, and they will flourish under the UV lights. You can see a bluish color there. Similarly, there are certain lasers which will allow you to see the fingerprints. Okay. So that's about it. और एक last चीज है आपका DNA fingerprinting and amylogenin gene. वो मैंने बहुत बार क्लास में भी कराया था आपको. So amylogenin gene, what is amylogenin gene? Can somebody quickly tell me? क्या होता है amylogenin gene? मैं ना पिटाई जाता नहीं है तो सुबह तक कैसे आएगा, अभी नहीं आता है तो.
मैम टूथ में होता है, एनामल फॉर्मेशन में हेल्प करता है.
Amylogenin gene, it is present on both X and Y chromosome. Okay, and it codes for the enamel of the tooth. Okay, but what is seen is that in the X chromosome it is 106 base pairs, in Y chromosomes it is 112 base pair. So when you do the STR analysis, STR analysis में क्या है, आप repeats देखते हो क्रोमोसोम्स पे, है ना. सो होमोलोगस क्रोमोसोम्स पे वो सेम ही आता है, लेकिन क्योंकि एक्स और वाई तो होमोलोगस नहीं है, सो x में अलग है और y में अलग. So x में वो 106 का है और y 112 का है. So you always see two peaks. Wherever you are seeing two peaks, let me show you. Just a minute. So amylogenin gene will help you to find out the sex of an individual. अगर किसी क्राइम सीन पे कोई सैंपल्स मिले हैं, अगर कोई सेक्सुअल असॉल्ट का क्राइम सीन है, उसमें कोई सैंपल्स मिलते हैं, कोई स्टेन्स मिलते हैं, और उसके बाद जब आप उसका एनालिसिस करते हो, यू सी, because a stain is a stain, you do not know what it will lead to. So when you do a DNA analysis of it, and you see two peaks. So ये ठीक नहीं है. Let me show you another one. Somewhat like just a minute. So somewhat like this, when you see two peaks, one shorter peak and one longer peak, that means the shorter one is from the X chromosome, the longer one is from the Y chromosome. और XY किस में होता है? मेल्स में होता है. अगर कोई फीमेल का होगा तो उसमें सिर्फ एक सिंगल पीक होगी, because they are homologous for the X chromosome. They will have only 106 बेस पेयर वाला ही, दोनों लोकस पे सेम जीन होगा 106 बेस पेयर्स पे. ठीक है. So what you see here, a male will have two peaks, one shorter, one longer. इसमें गलत बना है for the amylogenin gene, whereas a female will have only one peak for the X chromosome of 106 base pairs. ठीक है. जो STR एनालिसिस होता है उसमें ये पीक आपको क्यों दिखती है? प्लीज रीड DNA फिंगरप्रिंटिंग. ठीक है. पूछे जाएंगे आपसे वाइवा में, मैम आपको अच्छे से ग्रिल करेंगी. चलो, मुझे जो रिवाइज कराना था मैंने करा दिया. आई होप यू पीपल स्टडी एंड कम. किसी को कोई क्वेश्चन?
मैम परफॉर्म करने के लिए तो फिंगरप्रिंटिंग वाला एक्सपेरिमेंट आएगा ना?
परफॉर्म करने के लिए फिंगरप्रिंटिंग का एक्सपेरिमेंट आएगा, फिर की थ्योरी आएगी. यू शुड नो द patterns because you'll be asked to analyze them. So you should know what are the fingerprinting patterns. You should know what are the minutiae जो कि आप मार्क करते हैं, है ना. what are the methods of development, chemical methods, physical methods. फिंगरप्रिंटिंग से रिलेटेड जो भी आपने थ्योरी पढ़ी है वो सब आपका लिखने को आएगा ही आएगा. सो प्लीज स्टडी.
ओके मैम.
ठीक है. नहीं, न्यूमेरिकल ऐसे ऐसे क्वेश्चन पूछ लेते हो. न्यूमेरिकल अगर आएगा भी तो हम आपको दे देंगे उसका वैल्यू. मेरे पास ले आना निकाल के, मैं तुमको दे दूंगी उसका इन्वर्स करके. ठीक है. चलो जी, ऑल द बेस्ट टू ऑल ऑफ यू.
थैंक यू मैम.
ऑल द बेस्ट टू एवरीवन.
आप हो कॉल पे? ठीक है.
मैं अभी बीच में ज्वाइन करी हूँ. हाँ ठीक है.
ठीक है मैम. ओके मैम.
चलो.